All right. DNA has to do two things. We've already mentioned those. It has to be able to create you. You are protein. So DNA is the instructions for creating the stuff that you see, you touch. You are protein, okay? And DNA instructs how that is created. But the other thing DNA has to be able to do is copy itself exactly so you can tell your children how to be created. So your skin cell, this skin cell right here that says, okay, I need to make more skin cells, can tell its daughter skin cells exactly how to be skin cells, okay? So DNA must copy itself just to get its information to the next generation so they will be able to create themselves and so on and so on. So what we are focusing on in this is how copying occurs. Just copying that exact DNA replication. Okay? The basics of it are really simple and were kind of readily apparent once we figured out how DNA was built. Once scientists figured out that DNA had this complementary structure. If there's an A on this side, then there must be a T on the other strand. If there's a C over here, there must be a G over here. Then scientists began to imagine, well, if we unzip this strand, okay, which can easily be done because of those kind of weak hydrogen bonds, then perhaps an enzyme, we know everything in biology has to happen with the help of enzymes, just about. An enzyme would see A here and easily understand that T must go here. If C is here, then G must go here. If T is here, then A must go here. And same thing goes for this side. If there's a T, there must be an A. And so the replication process is just a matter of an enzyme bringing in the correct nucleotide to go opposite the already existing template strand. Okay, and that word template is important. The original parent strand acts as a template to help create the new strands that will be passed to the offspring. Okay, and so in the end, what you get are two completely identical strands of DNA, one here, and each of them is made up half of the old parent strand and half completely new DNA nucleotides. Half old, half completely new. And this one can go to one daughter cell and this can go to another daughter cell. And so we have replicated and passed on genetic information. This idea that newly replicated DNA is one half from the original parent and one half completely new is called being semi-conservative. The molecule is semi-conservative, meaning we conserve half of it. Half of it is the old, and we're going to keep that and add to it some new. Okay? All right. One more thing we need to understand and remember about DNA before we can talk about how it copies itself is that the molecule has a direction because each nucleotide has two different ends. Remember how we number this sugar. So here's the sugar, the nitrogenous base, and the phosphate group. The sugar has a carbon at each bend. A carbon is understood to be represented at each bend, plus there is a fifth carbon up here. And traditionally, they are numbered as the 1' carbon. So the first carbon after the oxygen is numbered 1. The next one is 2, then 3, then 4, and then the 5' carbon is up here. So there is a sense of direction. This carbon is different from anything else we see hanging off of these other carbons, and it always has the phosphate group hanging off of it. This carbon down here has a hydroxide group hanging off of it. These other carbons, the 1', the 2', the 4', we really don't care about. It is this 3' and 5' that we are going to keep a watch over because it will matter to us eventually. All right. There is a word that is going to come out of this 5', 3' directionality, and the word is anti-parallel. And that just means that two DNA molecules, I should have gotten this together before. This will work. Okay. Okay, I have two pens. Okay, the caps are here. In this case, the pens are running in the same direction. But if I turn one pen over so that its point is facing down and the other one's point is still facing up, they are now anti-parallel to each other. They are running in opposite directions. That's how the DNA molecule is as well. It runs in opposite directions each strand. Okay? We don't need to see that again. When we link nucleotides to make the long chain, phosphate, sugar, phosphate, sugar, phosphate, sugar, okay? We have the phosphate group attached to the 5'. Then there is this 3' carbon that is going to attach to the phosphate group of the next nucleotide in the chain. So we will have 5', 3', 5', 3', and another, the phosphate group here would attach to this, and then so on, okay? So this molecule has a 5' end and a 3' end, and the one opposite will have 5' and 3' ends as well, but they will be arranged upside down to each other. So this is the best drawing I can give you to show you that. If this is the 5' carbon here, its 3' counterpart is going to be down here, and if this molecule is going to hook up side by side with it, its 3' will be up here, and its 5' will be down here. They will be upside down relative to each other, anti-parallel. Okay? Now, why do we care? Why do I make a big deal out of this? Well, because that anti-parallel relationship is going to really affect how the enzymes are able to copy this molecule. Okay? The enzymes that copy DNA can only grow that DNA by hanging new nucleotides onto the 3' end of the carbon of the sugar. So I'm going to show you how that works in the next podcast. We'll pick up here next time. Thanks for watching. See you in the next podcast.